Thank you, Myron. It's a great pleasure to be here. And yes, as advertised, I'm going to be telling you about what a physicist thinks of animal development. And so, of course, the first question might be, what is animal development? And the basic question is, we have this egg here. And somehow or other, this egg knows what it wants to be when it grows up. But, you know, it's not very obvious. I can't tell from looking. So maybe this egg wants to be one of these, because, you know, that's a pretty cute animal, and who wouldn't want to be one of those? Or, you know, maybe it's been watching too many Geico ads, and it wants to be one of those. Or maybe, and I have it on good authority that this is the right answer, it wants to be a chicken. But, you know, really, it's not obvious from looking at the egg what it's going to turn into. Yet the egg knows, and it gets it right most of the time. You know, there are cases where there are birth defects or the equivalent in animals, but it's remarkably good at getting from something that looks nothing like a chicken over here to something that looks like a chicken over here. And so the big question I'm interested in is, how does biology know to do that, and how does biology know to get it right, at least most of the time? So this is and what interests me about this, or one reason that a physicist might get interested in this, is that this is a process of self-organization. So how does this not work? There is no miniature chicken inside the egg waiting to be blown up and become an adult chicken. I'm sure you all knew that, but for many centuries that's what people thought, and that made perfect sense, that how do you get a big chicken? You have a tiny little chicken sitting in there, and then it sort of inflates. Okay, but no, this was, you know, for centuries until 19th century or late 18th century, this is really what people thought. Okay, but we know that's not right. So maybe the way it works is this. And, you know, maybe there's a factory that makes the chickens. Now, of course, we all know that that's not true. But we should stop and think about this is actually a fairly profound statement that this is not true. The way we as humans know how to make things is with factories like this. We know how to make things by going in with our hands and putting them together, or maybe going in with a robot and putting them together, or, you know, having some external agent that assembles the thing the way we want it. And that's how we make essentially everything still today. But this is not what the chicken does. What the chicken does is it just has the egg, and the egg turns into the adult, and it pretty much doesn't get any help from anything else. It gets a little bit of help. Its mother tells it which end should be the head. But basically, it manages to sort of pull itself up by its own bootstraps and accomplish this amazing transformation and organize itself into this incredibly complex organism. Okay, and so how does it do that is then the obvious question. So one answer, and a question I get asked fairly often if I give this talk to a sort of a broader audience, is, well, haven't we all been told that there are genes or there's DNA that tells you that you are a person, and the chicken is a chicken, and a gecko is a gecko, and never the twain shall meet? And the answer is yes, absolutely. The DNA is important. You know, the chicken is the chicken DNA that says this, make this a chicken. But on the other hand, the DNA is really not enough. So there is not hidden inside the DNA in your cell nucleus a detailed blueprint that tells you where to put every feather, and tells you exactly how big to make the eye, and tells you how to, you know, space out the different fingers or toes on the feet, and so forth and so on. That, you know, even though there's a lot of information stored in your DNA, there's not nearly enough to have a complete 3D blueprint of what the animal would look like. And moreover, you know that identical twins can come out looking somewhat different depending on the environment they grow up in. So the DNA and the genes are not everything. They're the starting point. But what, in fact, they do is not specify every detail of how to make this chicken. Instead, what they do is they tell the chicken cells, and of course, like every animal, a chicken is made up of many individual cells, just as you are, just as everything else is. And these cells talk to each other, and they interact with each other. And it is through these conversations, through this communication, that they figure out among themselves how to organize themselves correctly. So here, for example, this is a picture of not a chicken wing, but a fruit fly wing, because this turns out to be much easier to take pictures of. And you can see that along the middle, there are some cells that look different. And these are going to become a vein. So, you know, same as veins in you that are carrying fluid and nutrients and stuff. And somehow, some cells have to decide to be the vein, and others have to decide not to be. And essentially, the way this works is that there's one cell that says, OK, I'm the vein cell. And it sends a signal to all of its neighbors saying, look, I'm the vein cell, and dude, don't mess with me. And the other cells say, OK, got the message, and I'll just be a wing cell. And of course, they don't do this with their voices. They do this by secreting chemicals, by sending out chemicals into the space between the cells that neighboring cells can then detect, and in some sense can smell, in the same way that maybe you've heard of pheromones are used to signal between ants or insects or stuff. They send out chemical signals, and different cells receive them, and in this way they talk to each other. And what the DNA is really telling you then is not exactly that this cell, that this one cell is going to be the vein cell and this other cell is not, but the rules for the conversation between these cells that will allow them to decide that really I only need one vein cell or a stripe one cell thick, and after that, the others should be wing cells. So what's important about this then is that there's nothing looking down over this, sort of pulling the strings and moving things around. There's no one cell that has a picture of the overall pattern. It happens all by fairly local communication among the cells. And this is really what we mean by self-organization. All right, so this seems fairly amazing that these things talk to each other and they spontaneously manage to come out the right way. That said, we do know of simpler physical systems where something like this happens. And so one of the simplest examples is a colloidal crystal. So this is a picture of a whole bunch of little styrofoam balls. They're quite small. They're fractions of a micron. And if you put these in, a container, a little dish or something, and maybe shake them up a little bit, they will, it's dense enough, they will spontaneously arrange themselves like this in this very ordered fashion. And they will do this despite the fact that, of course, none of these little styrofoam balls has a brain, and it is not thinking about where it is supposed to be. What it is doing is banging into its neighbors, and the forces from banging into its neighbors are allowing it to arrange itself in this crystal. And so you can imagine that if utterly mindless styrofoam balls can arrange themselves in some somewhat complex way, then cells that have an even better capacity to talk to each other might do even better. So it is plausible then that sort of local interactions and local conversations can bring you ordered patterns. And we know other examples in physical systems. So that was an example that is what we would call not driven. These are examples that are driven. That is, you have to put in energy to get these things. These are both patterns from convection, so from having sort of a layer of fluid that makes rolls as it goes up and down. And these are both chemical patterns. And we think we understand these things fairly well. And so just to convince you that I'm not making this up, that very simple systems can actually organize themselves into complex patterns, I have a little demonstration of that for you. And so I'm going to switch the video over, and with luck, we're going to get what we want out of that. And so, OK, so what I have here then is I have Yes, okay. So I have 4 vials of chemicals. And what I'm going to do is I'm going to mix them together. And what we're going to see is that they're going to turn into a nice pattern. And it's going to take a little while, so I'm going to sort of leave this up on some of the side screens and go on with the talk, and you can watch what this is doing. And you can see I'm putting on gloves because these are just slightly nasty chemicals. Not very bad, but bad enough that you would rather not get them on your hands. All right, so let us take these things and let us put them here on our little glass sheet. Okay, and you can see that this starts out as a red chemical, but we're going to see that it changes colors. And you can also see that it's hard to get everything out of these tiny little vials, which is why I'm tapping them. All right, and one more. All right, and so now what I'm going to do is I'm going to stir this up, and then we're going to sit back and watch what it does for a while. So give it a good stir. You can already see that it's starting to change colors. Yeah, so that's cool, and it gets better. Yeah, it does. All right, so we're going to just watch this thing, and I will come back and talk about how this is an example of self-organization in a fairly simple physical system. Okay, so while we watch what this does, let's go back and think about self-organization in biology. So making a chicken from an egg is a vastly more complex process than getting a bunch of little balls to line up correctly, or even than getting some chemicals to make a pattern. And you have to essentially do 3 things. You have to do what's called pattern formation or fate specification, and this means getting the right kind of cells in the right place. Then you have to do what's called morphogenesis, and this means once the cells know what they're supposed to be, they have to take on the correct shape and form, and a nerve cell should look like a nerve cell, and a liver cell should look like a liver cell. And I'm going to give examples of both of these, and then there's a third thing I'm not going to talk about today, which is how you decide how to stop growing and be the right size. But I want to start by talking about pattern formation. And so the basic goal here is to get the right cells in the right place. So you have a liver and you have a brain, and you really want to make sure that you put liver cells in the liver and brain cells in the brain. Right? It would turn out badly if you sort of mix those up some. And this is equally true on other smaller scales, that within the brain or the liver there are different kinds of cells, and they ought to be arranged in the correct way. And so we'd like to know how it does that. And there are a number of ideas out there about how it does that. And one of the most important ones is something called a Turing instability. And this involves what's known as an activator-inhibitor system. So A is activator, I is inhibitor. And you should think of these as 2 different chemical species, and I'm going to be sort of deliberately vague about what they are in more detail, because they can be many different things, and they have one identity in our chemical reaction and another identity in the actual in the animals I'm going to talk about. But what's important is that this activator A is able to cause the production of inhibitor I, and it's also able to cause the production of itself. So we have an activator that catalyzes the creation of more of itself. That's what this arrow that doubles back on itself says. And it catalyzes the production of the second thing that's the inhibitor. And then we have this inhibitor which blocks the creation of or slows down the creation of the activator. And we indicate that with a blunt-headed arrow and activation with a pointy-headed arrow. And So systems like this are fairly common in one way or another, and they're known to be able to make patterns. And the classic way they make patterns is something called a Turing instability. And basically what this amounts to is I start out with uniform activator in green, uniform inhibitor in red. And then this thing spontaneously decides that really it would rather be non-uniform. It would rather go and make something that looks like stripes. And the reason that works is that the inhibitor moves around faster than the activator. And so I can have this bump of green activator here. And if I have lots of activator, so we go back and remember that the activator is causing the creation of the inhibitor. If I have lots of activator in this green bump, then I should be making lots of inhibitor, and that could be a problem. But we can get away with it because the inhibitor moves faster, diffusion being the way it moves. And so this activator can sort of dump its inhibitor onto the neighboring cells, and thereby sustain a higher bump than it could sustain a constant concentration. So this is one way that activator-inhibitor systems work. And this is roughly what happens in our chemical reaction. And if we go back and look at the chemical reaction, which is not, there we are. If we go back and look at this chemical reaction, you can see that it's starting to make a pattern. And this pattern is going to continue to evolve, and it's going to start looking better. But you can sort of see over on the right that it's starting to make a series of stripes. Okay? And this is happening purely from the fact that there is one chemical in there, is the activator, that comes out blue. And this blue thing is in high concentration, and it's making red stuff. And then it's pushing the red stuff into the neighboring regions so that it can make a stripe where it's really blue. And this has a feature I didn't talk about in the pictures I just showed, which is that moreover it's making moving waves and it's growing. And this is also a common feature of activator-inhibitor systems. And so what this is meant to show us then is that with just 4 little chemicals in vials, I can make a nice pattern. So we have some hope of understanding how we make similar patterns in biology. So armed with this knowledge that we have ideas about how patterns can be made in physical systems, we went and we wanted to find examples of biological systems. And what we discovered, and so now I'm going to tell you a little bit about my own work in pattern formation. What we discovered is that these activator-inhibitor systems could work in multiple ways. And the way they appear often to work in biology is that this activator acts like a switch. So it makes more of itself. And what that means is if you have none of it, you can have a low concentration. That's one state. That's the off state, say. But then if you have lots of it, it keeps on making itself, and so it stays at a high concentration. And that would be the on state. And so the way biology is going to end up making a pattern is to sort of go around flipping these switches in certain cells via the communication between the cells. And we study this in a particular example. And this particular example is the fly's eye. And I should perhaps stop to say why flies. So beyond the fact that, of course, flies are wonderfully cool things. I mean, they're quite amazing. Flies are also easy to work with in the lab. They reproduce fast, so you can get new ones and get lots of mutants in a short time. And they're easy to breed, and moreover there's 100 years of history about how they work that helps us, that we can use as a starting point to try to understand them. So my collaborators and I were interested in how does biology make patterns? That is, how do we arrange to have the right kind of cells in the right place? And we went looking for a particular example where we could try to understand something. And then hopefully this example will give us broader insights. And that example is this fly's eye. And what you probably all know that flies have a compound eye. You've seen pictures of this or heard this at some point. What you may not have been told or may not have realized is that if you look at this compound eye and you look at the individual facets here, they're really arranged in an absolutely beautiful crystal. And so the fly started off life as a little egg. The egg did not have eyes. And so the fly had to somehow self-organize this crystal. It had to make this crystal for itself. And how does it do that? So it turns out that this starts out in something called the imaginal disk. This is a sheet of cells. So this is sort of like a sheet like your skin, only it's inside the fly larva. And it is going to turn into the adult eye. And what happens during the larval stage, so larva is the technical word for something like a caterpillar or grub, or the fly equivalent of that. And so what happens during the larval stage is that something called the morphogenetic furrow, which is this green stripe, starts at the back of the disc and moves across the disc from back to front. And ahead of the furrow you see no pattern. Behind it you can see a pattern. And in this case, this is a stain. That's the brown cells are going to become neurons and the non-brown cells aren't. And so what one might reasonably think then is that this pattern gets laid down at the moving front. And that is indeed known to be the case. And so if you go and blow up this green stripe, what you see is that at one end you sort of have low but uniform green expression. And then this green gene, atonal, so this gene, if a cell is green for the next few minutes, it means it's going to become what's called a photoreceptor, a cell that can detect light. And obviously you need photoreceptors in an eye, but you also need other things, cells to help out the photoreceptors. And so we see that we start out with sort of low uniform green, and we go through a process where we end up with reasonably spaced single green spots. And what's going to happen is that each of these single green spots, which is a pattern of expression of this gene atonal, is going to go and turn itself into one of these facets in the adult compound eye. So each little lens back here, so one lens there, say, corresponds to one green dot over here. So you get the green dots arranged correctly, or at least don't mess it up too badly, and you will end up with the right pattern of the adult eye. Okay, so what we want to know then is how do cells talk to each other to make this pattern of atonal expressing cells? And it turns out that a lot is known about this at the level of sort of circuit diagrams of how cells talk to each other. So it is known, for example, that this gene atonal, that if a cell has this gene turned on, it will favor making more of itself. That's what these pointed arrows mean. And it will favor making more of something else called senseless. And you can see that we have this whole fairly elaborate circuit diagram. And there are many, many decades of experiment that went into working out how these different pieces communicate with each other, and how if one cell makes scabrous, then this signal can go and tell another cell, turn off your atonal, and so forth and so on. But from a physicist's perspective, what this looks like is essentially one of these activator-inhibitor systems. So we have atonal that makes itself, we have atonal that makes scabrous, we have scabrous that inhibits atonal, and we have the same thing repeated several other times with other genes. So we started out by digging through the literature and drawing ourselves this sort of spaghetti diagram. And then we said, well, that's way too complicated for poor physicists. And so we thought we'd look at a simpler model, which essentially consists of an activator-inhibitor system plus a third activator that helps move this front that moves across the fly eye. Okay, and that's activation, that's an inhibition. You can turn this into equations. I'm not going to say anything more about that beyond that there are equations. But what's interesting is what we found out about how this works. And the way it works is with something we call switch and template pattern formation. And essentially what's going on is you have this, this long-range signal H that is moving forward here. And some cell sees this and it says, oh, I'm going to turn myself on. And so boom, it turns on A. And A, remember, is like a switch because A can activate itself, so it can stay low or it can stay high. So A switches itself on. And we get some stripe or region of instability, of expression. And then A starts making inhibitor. And this inhibitor tells the cells nearby, you better not turn yourselves on. So this is exactly the same communication we saw in the veins. One cell says, I have high A and I win, and none of you better turn on. And it says that by sending out a little signal in red to its neighbors. And its neighbors listen and say, okay, we won't turn on. But then after a while, the signal only goes so far. And the front can move on. And we can get another cell that starts to turn itself on. So a cartoon form then of the way our model works is that you imagine that you have two cells on here. There are these dots. And imagine that they tell everybody within a certain radius of them to turn off. So that's these circles. So we have two cells that are on, and they're going to on meaning they're like green dots, and they're going to found the space behind one of these lenses. And these green dots then are these dots send out an inhibitory signal and turn off everything around them. At the same time, there's a front moving forward telling everyone to turn on. And so the first place the next cell can turn on is in this little space here between the previous two circles. And so you can easily see how you fit a next column of cells, where's my pointer? There's my pointer. Here, in between the previous column. And this should give you a nice pattern. And then you can go on and you can put the next column of cells here and so forth and so on. All right, and this all works again by an activator inhibitor, but it works somewhat differently from the Turing instability I told you about. It's more like a race, that the first cell to turn on quickly sends out an I1 signal to its neighbors, says you stay off, and thereby you get nicely spaced cells that are on. So we can make a computer model of this, and this is what the computer model gives you. So what you're seeing here is a heat map. So red means on, dark blue means really off. Everything in between means something in between. And so what we're trying, the red here is the equivalent of the green cells in previous pictures. And we're trying to make a pattern of these red cells that stay on. And you can see that at least in my computer, I can take a simple idea of one gene tries to make more of itself, and then it sends out signals to its neighbors and says, don't you dare also turn yourself on. And this will go and make a very nice pattern. And you can see that these are each spaced, nice, equally spaced. So we get them, 3 of them, this one halfway between the previous 2, and so forth and so on. So this is very encouraging, and this suggests that we have some idea of how this pattern gets made in the fly. To really test it, we ought to go and see what happens. We ought to predict something and see what happens as a result of the prediction. And that turns out to be easy to do, because you can imagine, what if I started with a whole bunch of cells that are on, a whole stripe of cells that are on, instead of just single cells that are on. Then I would get the stripe of cells, and each one would be sending out inhibitory signals. And when I go to make my next column, I wouldn't know where to put the next column. So there's no clear space here where you can fit in the next cell, and that has to be where it goes. And instead, you expect then to make another stripe. And this is indeed what you can do in flies. So yeah, thank you. Better with less light. So if you look at this, what you see back here is a fairly nice pattern of single spots. And then what we did is we went and stopped the process for a while. In particular, stopped the cell's ability to communicate with each other and say, hey, you neighbor, turn off. And the result is that we get lots of cells that are on, that are lit up here. But then you can kick the process off again. And now you're doing the same thing, but you're starting off with something like a stripe. And what you can see is that it goes on and it makes more and more stripes. And so this is really wonderful from my perspective, because I started with a model about how different genes and different cells talk to each other. And I was actually able to predict that I could turn patterns of single cells into patterns of stripes. Okay, and so this suggests that we know something about how pattern formation is happening. That was not what I meant to do. Back to that. Right, what I meant to do was this. All right, and speaking of stripes, if we now go back and look at our reaction, you can see that this reaction has also gone and made stripes. So, self-organization in physics with chemicals that react with each other in sort of an activator-inhibitor way will make a stripe pattern. And similarly, biology will make a stripe pattern. And so, it's tremendously much more complex in detail what happens in biology. But in outline, the same ideas from a simple physical system, like these chemicals, will also let us understand a biological system. And unfortunately, we need to turn down the lights again every time I do this switch, like this fly, where I likewise get stripes when I do the right things to it. Okay, so I'm going to leave the reaction up on the side screen so that you can continue looking at it. So I started out saying that there were two things you need to do. You need pattern formation, that is, you need not to accidentally put liver cells in your brain or brain cells in your liver. But then once some cell knows it's a brain cell, it's sort of vaguely spherical, but it wants to end up like this. And this is not entirely the same shape as this. And so the second thing you need to do in order to organize yourself into this mythical adult chicken is you need to give cells the right shape, give cells and tissues the right shape. And this is called morphogenesis. And I want to show you a few examples of what we understand physically about how that happens. Okay, and I want to start with doing this at the tissue scale. And so here I want to talk about an example in flies that's called the ventral furrow. And basically what happens is that early on the fly egg is one layer of cells here. So all of this is yolk, no cells, and there's one layer of cells around the outside. And of course, an animal that's only one layer of cells thick is not a very good animal. So at some point, it has to decide to start moving some cells inside so that it can have not just skin, but also internal organs. And in fact, so the cells out here are going to become more or less the skin, and these cells that are going in are going to become the gut, the stomach and the intestines and stuff. And so you need to get from here to here. And this is just, so the bottom pictures are slices like this through the top picture showing you side on what's happening. And this is a view from the bottom of what's happened when you've invaginated. And I should also say here that in the second part, I'm going to show you quite a bit of data that is not mine. And is taken from various published papers of colleagues. And I want to be particularly careful to acknowledge them, and so I've gone and put the references over here. And so this is taken from a paper from Maria Lepton's group. Right, I just wanted to say that carefully because, as you may know, scientists tend to be very meticulous about giving each other credit. This is how we all get recognition and get ahead in the world. So these are some very nice pictures from Maria Lepton's lab. And I'm now going to show you some movies that come from the lab of Wayne Broadland and Buzz Baum that are about the same process. So, no, I'm not going to show you the movies next. Okay, so I'm going to show you the demos next. So let me go back and say a bit more about this. So I have this black tissue here, and I want to get it to go from more or less flat to bulging in. Okay? And you can imagine two ways of doing that. You might, or at least you can easily see physical examples of two ways of doing that. And we have sort of a little physical demo here to try to show that to you. So we're going to imagine that this ruler is not a ruler, but is a set of cells. That's sort of, this is the bottom of the fly. And we now want to make it bow inwards. And you can imagine different ways you could exert physical forces on it. You could push and pull on it in order to get it to do that. One is you could push in from the ends. And I'm sure all of you have done this experiment yourself at home. You push and, maybe not even intentionally. You push and at some point it buckles inward. So that's an easy way to get something to buckle in. If you're a cell, you might also be able to do a different way, which is and okay, I want to screw this back in and be very careful it's tight, which is the cells in here, which I'm now representing as rubber tubing, might be able to do their own pulling in the middle. Okay. And then what you would get is you would get something more or less, so like this. So again, you would get that, okay, so good. So again, you would get that you've bowed in. And if I pulled even harder on this, I would bow in even more. Okay, so it's obviously very important to the embryo to get its similar bowing process to work right, to get these cells here to bow inwards. And again, it needs to have some way to decide to do that. And I should say, incidentally, that pattern formation has already happened. So there's already been communication between these cells over here and the black ones down here that has led the black ones to decide that we're the ones that are going to try to bow inwards, and the other ones to decide we're not the ones that are going to try to bow inwards. So we're now on to the second half of the question, as I said, which is how did these cells actually manage to push and pull on themselves to go get to there? Okay, and I just showed you two mechanisms. So this group, the Brodlin group, wanted to try to investigate that. And so here this is a movie of that bowing process. So the cells bow in, and then we get here this tube that is going to turn into the gut. Let's watch that one more time. And they set about to investigate this, and it's not easy to know what is pulling on what in a living embryo. So one thing you can do is you can make guesses about what is pulling on what, and you can then try to make a computer version of this embryo and see if it acts the same. And that's what the Brodlin group did. And so here is a computer model of the same thing, which has made some guess. And the guess here is that what's important is that these cells here are acting like the rubber tubing. That is, they are pulling in on their tops, and that is causing the whole thing to bend in and go and so make this invagination. So the proposal then is that what's important is to have the cells that end up in the middle here really pull their tops in very tightly and end up and act like the rubber tubing. And that pushing from these cells here on the side is less important. And one way to test that is to remove the ability of the cells to pull in, which you can do by getting an appropriate mutant, and this is such a mutant. And you can see that it doesn't bow in. And eventually it gets confused and tries to move in other places. But the first thing that happens is it starts out looking pretty okay, and then it doesn't bow itself in. So all right, so clearly pulling from the top then is important in invagination. And you can again test your computer model. And there we go. There we go, okay. Right, and so the point is precisely that not much is happening here. So this is a model of cells where the ability of these ends to contract as the rubber tubing contracted has been taken away. And again, nothing happens. All right, so here's an example where we wanted to know how does biology organize itself to push itself in and make a tube. And we think that what it is that it's pulling itself from the middle, not having external forces push it in. You can also ask about morphogenesis, about how things get their shape at a different scale, that is the scale of how individual cells get their shape. And so this is taken from a now fairly famous paper by Rich Carthew's group. And these are cells that are on the inside of the fly eye. So this is later on close to the adult eye. And sort of each of these circles here, these circular collections of cells, goes behind one lens. And so what Carthew and collaborators asked is what determines exactly the shape of these 4 cells in the middle. And in normal flies, it's always 4 cells, but they have mutants where it's sometimes 5. And it sort of looked to them like soap bubbles. And so they went and did a very elegant experiment where they made collections of soap bubbles, which is in the left column here, with a certain number of bubbles. And then they went and they found collections of cells in the right column here that behave similar, that have the same number of cells. And then they went and compared the shapes. And what you can see is that the shapes of the aggregates look really amazingly similar. So a single cell is circular, that's maybe not such a shock. But when you get over to, say, 5 cells, you see that it arranges itself in this particular way with 3 and 2, and 3 and 2 over here. And you know, there's no obvious reason to do that. And same thing with 6 cells, you get 6 cells like that, and so forth. So the suggestion then is that at the scale of single cells, one thing that is very important in determining their shape is surface tension. And some of you may remember surface tension from Robert Deegan's Saturday morning physics talk, I believe earlier this semester when he talked about capillarity. And so the basic idea is that if I have a droplet or a bubble like this, every little bit of it is pulling outwards on every other bit. So these arrows indicate that there's this little black bit there, and its neighbor over here is pulling it that way, and its neighbor over here is pulling it that way. These forces arrange and shape the cell. And what we believe is that cells control these forces and thereby control how they're shaped. And so one thing we can do is we can go look at embryos and we can try to figure out from pictures of embryos what these forces are. And it turns out that there's a very simple idea here, which is if you imagine, so I'm going slightly ahead of myself to show you a picture of the actual system. So here are some cells, and these are the edges of the cell. So this is 3 cells. So usually the black cursor is good, but in this particular picture it's not so good. So here are 3 cells, and here are their edges. And what we want to know is, given the way their edges are arranged, what can we figure out about who's pulling hardest on whom? And it turns out that there's simple physical ideas that allow you to do this. And so the basic idea is that suppose that I have 3 edges that are pulling equally hard. Then I expect, then to balance themselves out, I should get something like 120-degree angles. So they should look equally, equally spaced, because they're all pulling equally hard. And so if they're going to balance out, they should space themselves out nicely in this fashion. On the other hand, if I see something like this, where 2 edges are almost parallel and a third one is going up, this means that these 2 edges are pulling much harder. And a third edge, and they're mostly just canceling each other out. And then the third edge is not doing very much. And so we have a demo here that tries to make this point. So this is fairly low tech, but it gets the job done. So here we have, these are 3 different scales. They're spring scales. This is what you would have, this is what holds the basket that weighs your fruit in the supermarket, for example. And we've started out with a configuration where you can see that the force on these 3 scales is roughly the same. So that means each of these 3 yellow wires is pushing roughly equally hard, or pulling rather roughly equally hard. And correspondingly, we have roughly equal angles. On the other hand, I could go and move these things. And so I could say take these and move them down and out. So the blackboard moves on me too. OK, and so now what we have is we have not remotely 120-degree angles. So these 2 are almost parallel, and this one is coming down like that. And what you can see is that this dial and this dial read a force that's the opposite of the, so the pink is telling you the top of the dial. And so this is down at 6 o'clock. This one is down at almost 6 o'clock. And this one is at sort of 2 or 3 o'clock. So then if I see a configuration where I have two lines that come in almost together and a third one that comes in perpendicular, this means that these two lines are pulling harder than this one. Conversely, if I see a situation where these two are, okay, so yeah, that works. These even more too. Good. Okay, so conversely, if I see a situation where I have these two fairly close together and this one straight down, you can see that now both of these are at around 5 o'clock and 5 o'clock, and this one over here is at something like 7 o'clock. And I can probably actually make it even better by moving it down a bit. There. Hello. Now, okay. Now it's stuck. Oh no, it's past. I see. Okay. That's what's going on. All right. There we go. That's what I wanted to show you. Sorry. These are strong magnets. They have. All right. So this one. So V here, small angle. This one, big force, almost back to 12 o'clock. So something like 11 o'clock. All right. So then the idea here again is that by looking at angles, we can infer how hard things are pulling on each other. And in particular, it's very interesting if things are not every edge is pulling equally hard, because that tells you something about how cells are trying to organize themselves. All right, and you can see, and this is a picture from work I've done on fish, that here, for example, I have basically a straight line, something coming in perpendicular. And that means that there's a very high tension along the vertical and a very weak tension along the horizontal. And you can, again, take a computer and simulate what this does. So here we're going to be pulling hard along edges, edges between cells that are vertical and not hard along the horizontal ones. And what you see is that this makes very straight lines of cells vertically, because let me stop it there. So along here we're pulling hard, and along here horizontally we're not, and correspondingly we have a junction here where this is coming in almost to 90 degrees and these are almost straight. All right. So why might you want to do this? Well, one reason is to get cells to line up like this in columns. But there are other reasons. So, and these are movies that do not embed well in my PowerPoint, so I'm going to go out of my PowerPoint and show you them this way. So there's a process called convergent extension. So the idea is that we have these, and this is again a computer model, and I will show you some data in a moment. We have this block of tissue, which it turns out is sitting around the outside of the fly embryo. And it wants to squish itself down vertically and get longer horizontally. That's its job. That's how it's supposed to rearrange itself to get to the final shape it needs. And what this model suggests is that the way it does it is that on the red edges, you really pull down hard. And on the black edges, you don't. And this way, you compress it vertically and allow it to spread out horizontally. And in fact, you get the cells to intercalate between each other. And so here is, okay. And these three differ by how hard you're pulling down vertically relative to horizontally. So this is the weakest difference. This is the strongest difference. Let's show that again. And you can see that here, where there's a very strong difference, it elongates the most. All right. So, so these people, and this is the group of Thomas Lepie, hypothesized then that in this fly tissue, you would go, that if you went in and measured the tensions, what you would see is that the vertical edges have very high tension, and the horizontal edges have very low tension. And this way you get from there, which is a somewhat anisotropic rectangle, but not too bad, to there where you're really elongated. And the question is, how do you actually then go test that this idea is right? And it turns out that the idea is fairly simple. It's basically that you go and you zap the thing as hard as you can with a laser, and then you watch how it reacts. And I'm not going to zap a fly with a laser for you, but I'm going to do the same thing with the soap film. And it works more or less the same way. So what I have here is I have a, so we arrange so we can see this. All right, so I have a frame here, and I'm going to put a soap film over it. And then I'm going to break part of it, and we're going to watch the rest of the soap film pull this little wire around. Okay? And this, firstly, will show us that the soap film really is pulling, that there really is surface tension. And then in a second, I'll show you how it can, moreover, let us measure something about the surface tension. So first off, let's do the first experiment. Lost the film before I could do the experiment. All right. Try that again. All right. So here we go. And wow, you can see it zips around. All right. So that was so much fun. We should try it again because it also went pretty fast. All right. This is not working. Okay, so I'm having trouble. That was not the goal. All right, I think I'm going to leave them to fix the demo since the wire fell out, and go continue talking for a moment. And maybe actually this would be a good moment to go back and look at the side screens. And you can see that our self-organization experiment is going on. And you can see that we have some very nice stripes there. Okay? So this again returns to the idea that chemicals activating and inhibiting each other can go and make stripes. All right. On the other hand, you can, are we good to go? All right, then I won't have to go back. Yes, I've been doing that. All right, so let's watch this again. There we go. Okay, so we saw that there was a force, and we saw that that thing went around really fast. So what's interesting, though, is we could try it with some other material. And so this is a different soap solution that has somewhat different properties. And we can go and look at what happens with this other soap solution. All right, so here we have a different soap solution. And you can see it went more slowly. In fact, it got stuck. So yeah, about that. Let us again try this one again, see if I can get a soap film. There we go. All right. So there, goes around slowly. So what does this tell us? This tells us that by breaking something and then watching how it recoils, how other things move, we can learn something. And so what we learned here was that apparently one of these is pulling harder on the little wire than the other one, or something else is causing one of these to move more slowly than the other. In this case, the biggest difference is that one of these has fluid that's much more viscous than the other. So basically there was the equivalent of honey inside the second one, so it went more slowly. If in flies, we don't have honey in some cells and water in other cells, but you can do the equivalent thing. You can go and zap something, and you can watch how it retracts. And from this, you can try to figure out how hard it's pulling. And so, wait and come back to the, right, I was supposed to be doing that. That's the problem. Okay. There we go. Okay. So here's a movie where we're going to go and we're going to, so we have these cells, and we think that there's tension along all of these lines joining the cells. And so what we're going to do is we're going to take a laser and go in and zap one of these interfaces. This is more or less the same technology that is involved in LASIK surgery, incidentally. So this is not something so incredibly exotic. You all have some idea that you can use lasers to cut things. And let's look at what happens. And there's zap, and it recoils. And you can measure how fast it recoils. And by doing that, you can try to figure out how fast it's, how strong the tension is. So we can watch it again. There's zap and recoil. All right. And so you can do this many times. And I just want to look at particularly this graph. And what you can see, and this is as a function of the angle it makes with the vertical, how fast does it retract? And what you can see is that these lines go up. So what this is saying is that, or sorry, this is the angle it makes with the horizontal. What this is saying is that vertical ones retract much faster, and correspondingly they have much more tension on them. And so this brings us back to the idea that the vertical ones are pulling hard, the horizontal ones are not pulling hard, and in this way you can get your rectangle to squish down one way and elongate the other way. All right. So with that, all right. So this is what I had prepared for you today, and timing is pretty good. So what I wanted you to get out of this then is that development is really a process of self-organization. And that is, the chicken makes itself. And we have physical ideas about how self-organization can happen. For example, in this chemical activator-inhibitor system. And we also have ideas about how cell shapes can be set by either pushing down on a ruler or pulling its middle with a tube, and about how cells can rearrange themselves in their shapes by having different tensions. And so we can try to start to understand this self-organization process. And we're nowhere near understanding all of it. Biology is incredibly complicated, and we will not in my lifetime know everything about how eggs turn into chickens. But we're starting to learn something. And so I want to conclude then by saying something about why are we doing this and what do we hope to get out of it. So my biggest motivation is just to understand how biology works, because it's pretty amazing that you start from a chicken and you end up, or start from an egg and you end up with a chicken. And this also has some medical relevance because, of course, when this process goes wrong, you get the equivalent of birth defects. But the other thing about this is that, so biology is clearly extremely good at self-organizing. And we would love to be able to do that. We would love to be able to have things that spontaneously make themselves into what we want them to be, instead of our having to have that assembly line I showed you a picture of. And so the other direction that this research goes is towards learning from biology how to make man-made, non-living systems that similarly self-organize. So learning from the principles of the biology that what principles we might apply to man-made systems. And indeed, the way people thought about making this chemical reaction system that's making these stripes is precisely that they had this idea that there were activators and inhibitors in biology, and they went and tried to find some chemicals that would do the same thing. Okay, and with that, I would like to thank a few people. All right, and with that, I'll stop, and I think we have a pause, and then I'll be happy to ask questions.